Right, so we're going to move on to translation. So now we have the messenger RNA that was created in transcription. And now, as the messenger RNA leaves the nucleus, it's going to be translated to create your proteins. So this occurs on ribosomes. Ribosomes are an organelle found in eukaryotic cells. So, how does translation work? So, the first thing that happens is your messenger RNA is going to give us the code to build our proteins. So, translations always starts with a start codon, and a codon is consisted of three RNA bases, and the start codon here is called AUG. So, this signifies the start of translation. So our start codon is going to be binded directly to our transfer RNA. So if you take a look at this picture here, this is our transfer RNA. Transfer RNA is attached to an amino acid. So in order to figure out which amino acid fits directly here, we look at the anticodon of the transfer RNA. So we can see that the anticodon UAC base pairs correctly with our AUG start codon. In this example here, our transfer RNA with the alanine amino acid is going to base pair directly with the mRNA here. So our transfer RNA with the amino acid attached is going to bind directly to the start codon of the messenger RNA. So this is going to continue, and you're going to end up forming an amino acid chain. This is called a polypeptide, which is going to be called a protein. So essentially, proteins are made up of amino acids. So each transfer RNA is going to come to the messenger RNA. And then once it already attaches, it's going to continue building the polypeptide chain. And we know that translation stops when we reach a stop codon. So whenever we have a stop codon, there's no amino acid for it. So stop means stop. You don't even need to write the word stop. It just means that translation is officially over. So we're going to take a look at how this works. So messenger RNA is a long code. Every three RNA bases is called a codon. So every codon will code for a specific amino acid. In this picture, we have three codons, which means that this is going to be an amino acid chain that consists of three amino acids. So a little bit of review. You have your DNA. From your DNA, we create our messenger RNA through the process of transcription, and this occurs in the nucleus of the cell. Then our messenger RNA is going to leave the nucleus and go into the ribosomes, another organelle in the cell. And every three RNA bases, called a codon, is used to connect to an amino acid. So every codon will give you one amino acid. So, a little bit of review, the process of what creates messenger RNA from what. So, this one's the process of transcription creates messenger RNA from DNA. The process of transcription occurs where in the cell? The process of transcription occurs in the nucleus of the cell. The process of blank uses the messenger RNA to create. So here we're going through the process of translation to use the messenger RNA to create our amino acids. And amino acids make up proteins. And lastly, the process of translation occurs in the ribosomes. So specifically on the ribosomes found in the cell. All right, so just to go over how this works. So earlier in the transcription lesson, we talked about how to create the messenger RNA when given the coding DNA. So first thing you do is you create the 3' to 5' template DNA strand using base pairing rules. So A with T, A with T, T with A, etc. Then you use the template DNA strand to create your messenger RNA strand. So like A with T, A with T, U with A, etc. So keep in mind your messenger RNA has a U instead of a T. Now, how do we go from messenger RNA to our amino acid chain? So every codon is going to code for a different amino acid. So we're going to look at how that works. You never have to memorize this chart. Do not ever copy it. You'll never in your life have to memorize this chart. So if we have the codon UCG, I'm going to use this chart. So I see the first base U. And then from here, I'm going to see which of these letters is my second base. And it says C. So I'm going to follow here. And then lastly, which of these letters is my third base? G. So that gives me serine. ACA. So we start here, we have A, then we go to our second base, which is a C, and then A. So this gives us the amino acid threonine. Then we have GGA. So starting from G, then we go to G here, and then A. So that gives us glycine. And last one, U. Then we reach G, and then A. Oh, stop. So every time you hit stop, it means stop. Translation is over. You do not need to write the word stop. So from our messenger RNA strand, every 3 RNA bases makes up a codon. I'm going to use the chart. So AAU, what does AAU give me? Asparagine. So we get asparagine. You can check my work, but GAC will give us aspartic acid and GAU will also give us aspartic acid. So this messenger RNA that contained 9 bases is going to give us a total of 3 amino acids.